Okay, so I'm just testing the microphone now. Can someone give me a thumbs up to say that it's working? Okay, just trying this out. Anyone, is this working? Hello, hello. I genuinely cannot tell. Help, help.
Yeah.
Could you test it? Could you test it?
Yep, I can test it.
Oh, it's really good, yeah. Okay, you're just doing it longer.
Yeah, I'll just put the I'm gonna check for like certainly.
Because I'm leaving like I might leave soon. So I'll talk to you 30. So probably don't want to. I just want to make sure everything's good. Set up.
So it was like just really like, we like rolled it at like 2 o'clock in the morning and go to bed, like and still be squeaking outside at like 5:30. So I was pretty tired by the time I got back as well as everything. Yeah, but yeah, it was good. It was good, good kind of good science, good catching up with people. Stephanie and we still lots of attractive posters, so it was good. Yeah, it was nice to be out with some of the almost immediate lights. Yeah. So, yeah. It was a nice location. It's like, you know, if you walk down to the waterfront for lunch and stuff like that. Yeah. Nice to feel like outside. Yeah. It's okay, your accommodation. No. So you still go to Florida or yeah, we ended up literally so much very exciting. I mean, she didn't have any children at that stage. She has like you. So yeah, so my mum's kind of in order to not so that it was only out there to expensive because we sit here for a little while. So I was actually, you guys might be on my rotation list of asking who's going to be around London at Christmas time. Really, because I'm like, pop in and feed the cats at various points. So I need to find a bunch of people. You know, Stephanie might go to person. So. Sorry. All right. Coming on. Oh, God. Right. Right. Okay. Well, three things. Like roll this out, like keep the feet
Are you making the?
I don't know. I'm not supposed to be in charge, but I'm taking pictures of the place.
Okay, I'll ask Natasha. Hey, how's it going?
Good.
Okay. So you either, I guess you can sit, but like feel free to walk around like the perimeter because you're like the photography, right? So like feel free to post and like make like 1 or 2 stories right now, just so it's like people can see.
Yeah.
Like maybe like one now before kind of everything starts, and then maybe one during, and then just take pictures. And like I know Samantha's also taking pictures, so like hopefully like, everybody just takes a bunch of pictures and we can like edit that together some way afterwards. But lots of people, looks good, right?
Yeah.
Not bad. And it's not even started. Have you ever done it?
Yeah.
At the back, because nobody's gonna be looking back. Only Dr. Sinclair is gonna be looking at you. So, and he's like a pro.
Unless if there's anyone else coming, if there's just trickle you in slowly.
Yeah, maybe give it like a CO2, like 1 or 2 minutes.
Yeah.
Oh, you just use the pen. That's a good number.
Yeah. Thanks. Yeah.
Hello, can someone type if you can hear me on Zoom?
Yep, we can hear you.
Hello? Hello, can someone type if you can? Okay, thank you. Hi, everyone. I think we are going to get started now. So good afternoon, everyone. My name is Sam, and I'm joining you as the co-VP outreach of the Biology Undergraduate Society. I'm so excited to see that there are so many of you joining us this afternoon, online and in person. So before we start, I would like to spend a minute to talk about Dr. Helen Battle, after whom this lecture is named in honor of. Dr. Helen Battle is a graduate of Western University and the first woman in Canada to earn a PhD in marine biology, after which she joined the Western faculty and began a teaching career spanning 50 years and 4,500 students. During her tenure, she was a vocal advocate for women in higher education and the improvement of the treatment of women at Western University. We at the Biology Undergraduate Society hope to continue her legacy of scientific education and advocacy by hosting a semesterly research and inviting guest speakers from our own biology department. Sorry, this semester, Dr. Sinclair has graciously taken time out of his busy schedule to tell us about the role of undergraduate research here at Western and a little bit about his current field of research. Please welcome Dr. Sinclair.
Cool. Thanks, Sam. And thank you for inviting me to do this. I'll just get to a point where I can see things here. I hope everyone can hear me online. If you can't, then send a chat message to Sam and he'll give me a wave and I'll change the position of the microphone. I apologize, I don't have the camera on for those of you that aren't here, but I assure you that I'm devastatingly handsome. So, oh, I was expecting something to happen there. Let's see if something happens now. Now something happens. Okay, so I just wanted to begin with a land acknowledgement, and we read these words and we see them and you can read them for yourselves. But I also want to take the time to just talk a little bit about the fact that in my lab, we study winter and we study the effects of winter on biology. And this is something that Indigenous people in this area were very aware of. It's a very, very important selection event for the plants and animals in the area and for the people living here. And so we have a lot to learn from Indigenous people and people that have lived traditional lives and traditional ways of life in these kinds of environments. And it's important to be aware of that, to recognize the ongoing injustices, and also to each do our part in reconciliation. And that goes beyond just putting words on a slide. It needs to include supporting and encouraging actual action. We've already talked a little bit about Helen Battle, and I'll see what I covered that Sam didn't. So she was born in London. She started her undergrad at 16. So those of you that are undergrads, who was older than 16 when you started your undergrad degree? So she was pretty young, and so that meant that she finished her degree pretty quickly. She rocketed into a pretty solid career. She did her bachelor's and master's here, and then her PhD at U of T. She was a professor here from 1929 to 1967, and she did a whole bunch of cool things. In my part of the world, we know her because she was one of the co-founders of the Canadian Society of Zoologists. She was the second president of the Canadian Society of Zoologists, and she was the first female recipient of the Fry Medal, which is the Lifetime Achievement Award in zoology in Canada. So she really had an enormous impact. And from a scientific perspective, she was one of the first people to actually use fish embryos to do ecotoxicology. So she started an industry, sort of a cottage industry that still exists, where we're using fish embryos, zebrafish these days, to understand the effects of pollutants in the aquatic environment. Okay. So I should apologize in advance, especially to my colleagues who have come here possibly in the expectation that they might learn something about what I pretend is my research, because I'm going to do a little bit of a bait and switch here, in that my take-home message is here, which is that undergraduate research is real research. And so what I really want to focus on is the role of undergrads in research and how you as undergrads can contribute to the research and contribute to the scientific endeavor. And you'll see how I make that argument as we go. I will talk a little bit about my research, but my real focus is about undergrads and what you can do. And I want to start by talking about two probably of the most successful biology honors thesis projects of all time. So the first is this guy here, a guy called Don Griffin. And when he was an undergrad at Harvard, he was the person that worked out that bats can echolocate and that they use sound to do it. And he did this as an undergrad and then built a career around it and really became like kind of Captain Bat for much of the 20th century. The other person is Marla Sokolowski. She's a prof at University of Toronto. And when she was an undergrad, she was like watching Drosophila maggots slurp around on petri dishes. And she noticed that some of them kind of stay still and some of them kind of wander around. And then for her honors project, she showed that that could be inherited in a Mendelian fashion. She found that it was this forager gene that had a rover genotype and a sitter genotype. And Marla basically took this and ran with it for her entire career and this is like one of the prime examples of a single gene determining behavior and has led to all sorts of understanding about cell biology and about and about how genes influence behavior. So a couple of famous people. Why were they successful? Well, they were both interested in biology. That's kind of important if you're going to be a biologist. But both of these things were associated with personal observations. They were directly involved in these things and they understood the literature context. You know, Don Griffin knew, in fact, that a guy Spallanzani in the 18th century had kind of suggested that maybe bats could see with their ears. And Marla knew that genes for behaviors were rare and important. They identified this novel finding and then they pursued it. And I'm guessing that they also worked pretty hard and had quite a lot of perseverance. And I'll talk about perseverance that I've observed in some of our undergrads as we go through. Now I'm going to talk, I'm not Don Griffin or Marla Sokolowski, but I'm going to talk a little bit about my origin story because in some ways it's a little bit similar in that I'm still kind of doing the same things now that I was doing when I was an honors student. And so my origin story as an undergrad was that I wasn't really sure what I wanted to do. In fact, I actually wanted to be an opera singer when I was 16. But when I was in second year, we had a kind of a mandatory ecology field trip to an island called Tiritiri Matangi. Oh, I didn't mention, I'm from New Zealand. That's why I talk funny. Small island, South Pacific. And we have all these nice islands in the harbor. And we went out there and we wandered around for a few days. The weather was not like this. We wandered around in the rain and did some kind of like make work project where we followed some bird and figured out how much time it spent doing different things. And I was like, this is amazing. This is what I want to do. I want to run around in the bush and chase birds. So that summer, I managed to weasel my way into a research job with these people, Mick Clout, Shane Walker or Shane Wright, sorry, and Astrid Dijkgraaf. And I did a bunch of stuff around Auckland where I grew up, and including going to this island, Hauturu, Little Barrier Island, which at the time was very hard to get to because it had all sorts of rare species and it didn't have some of the invasive species that have reduced biodiversity elsewhere in New Zealand. And so this was amazing. And one of the things that was amazing there is that Hauturu is the home of the heaviest insect in the world. So Deinacrita heteracantha, it's a giant weta. And they're basically a big grasshopper that weighs about the same amount as an American robin, about 70 grams. They're really huge. And these were kind of approximately everywhere. You could wander around and see them. And I was like, this is amazing. Insects are kind of cool. So few things happened and I ended up at Otago University in my third year. And we had these little independent research projects. And one that I was sort of assigned to was studying these little, you can barely see it. This is not a giant weta. This is a perfectly normal sized sort of small cricket called Isoplectron armatum. And we were figuring out what they were doing in their little burrows and things. And it involved like, you know, going out at night and counting weta and things. And that was pretty interesting. And I was also, having gotten excited about running around in the bush chasing birds, I was trying to kind of get myself involved in anything else that I could. And so I ended up volunteering on a bunch of different things. So I did a bunch of work on subalpine geckos with Alison Cree and Jenny Rock there. Jenny's now actually a professor at Otago University. And I also, Otago's nicely placed. There's people out doing things all the time. So I got to go and chase seabirds and measure plants and stand in streams and all of those things. And none of these were kind of, you know, posted on the notice boards as something that I could do. I just kind of like figured out who was doing stuff and kind of knock on the door and say, so can I come and help, you know, handle your albatross or whatever? But a big shift for me was in a third year physiology class, third year environmental physiology. I would say the equivalent of biology 3601 or 3602, those of you that are taking those courses. And it's still being run. I did this course in 1995 and it's still being run in 2022 and 2023. I looked it up on the website. And the thing that was a real revelation for me was actually a guest lecture by this guy here, a guy called Bill Block. And Bill was visiting on sabbatical. He was a researcher at the British Antarctic Survey in Cambridge. And he gave a talk about insects and insects at low temperatures. And what he told us about was that there are two main strategies for being a very cold tolerant ectotherm, right? So an ectotherm is an animal whose body temperature is pretty similar to the environmental temperature. And if it's very cold, then you run the problem that you run the risk of freezing. So, especially when it's, say -20, if your body fluids freeze at -3 or -5, then you really run the risk of ice formation. And there's two main strategies to survive this. So one is to be freeze avoidant, like these creatures here. And what these things do is they depress the temperature that they freeze at. So they accumulate often cryoprotectants, things like glycerol and things. They might use antifreeze proteins to stop ice from growing in the first place. And you can keep these guys liquid at really low temperatures. In fact, this is an emerald ash borer. They actually overwinter as really ugly pre-pupae, so I show the nice beetle instead. And these guys survive over winter. And in London, when we first studied them, they survived down to about -29, -30 degrees C, which was pretty cold, but not super cold. So we were like, oh, this is cool. So we did a bunch of modeling and we wrote this great paper about how we didn't think that they'd survive in very cold places like Winnipeg, Manitoba. And literally the same week that the paper came out saying that we didn't think that they would survive in Winnipeg, Manitoba, there was a big news story in CBC on the CBC website about how emerald ash borer had been found in Winnipeg, Manitoba. So we sort of said, oh, that's a little bit embarrassing. Can we, have some? And we got our hands on some of these beetles, and the beetles from Winnipeg, Manitoba can literally survive temperatures as low as -50 degrees C. So there are by the time you're inside a tree, there are no temperatures in Canada where you can grow a tree and kill emerald ash borer. So they're essentially going to live anywhere in North America that they want to live. So just as a reminder, these guys are freeze avoidant. They survive as long as they don't freeze. And if they do freeze, they die. And then the thing that really got my heart racing was learning about these guys, which are things that are freeze tolerant. So this is a species we get in New Zealand. This is another species of weta, another species of these kind of grasshopper things. And they live in the alpine zone. This is a woolly bear caterpillar. You will have seen them crossing your path over the course of the fall around here. And then we have a Eurosta solidaginus, the goldenrod gallfly, and you'll see the galls on goldenrod plants and kind of weedy fields and things like that. So these things are all freeze tolerant. So this means that when they cool down, ice starts to form and they freeze solid. If you drop them, they will break. If you do not drop them and you thaw them out again, then they will walk off again. Or in the case of Eurosta, just kind of sit there like a pointless blob anyway. And I was like, this is amazing. That is what I want to do, right? And I was very much in luck, and I can't emphasize enough how much luck plays into how any of us end up doing what we do in science and in life. And in this case, the thing was that Bill was visiting and was giving a guest lecture, and Dave Wharton, who was actually a nematode physiologist, he ended up being my PhD advisor. He was the prof that was teaching the course. And he also had visiting him Hans Ramlov, a Danish expert on antifreeze proteins. And I was like, I really want to do research on this. And they're like, well, come along. And so they took me on a trip up into the mountains. We sat around and talked about science and insects and freezing and what we didn't know. And not long after that, another guy from British Antarctic Survey, so Roger Warland, came to visit as well. And by pure chance, as like the one like undergrad who was interested in this stuff and had like kind of knocked on the door of the lab, I got immersed in this really involved group of people that were doing research on insects and cold. And I also got like really interested in the topic, and I actually spent essentially the break between semesters in the library reading about insect cold tolerance and reading all the papers I could possibly find about it. And completely as it happened, that gave me enough of a background that I ended up being able to actually use it to write a review, which took a while to get published, but I ended up writing an article based on my my between semesters research as an undergrad, which has been cited more than 200 times. So it's actually not terrible. So on the back of this, I signed up to do an honors thesis, right? So good thing. Who's doing 4999? One person. There's only one 4999 person here. Two. Oh, but they let you in, Kathleen? I thought you had to have a good average for that. Oh, right. Yeah, you still haven't passed. Yeah, that happens. Yeah. It's a good thing. They've never asked to see my transcript to work here. Have they ever asked to see yours? Yeah, see. So signed up to do an honors thesis. I was super excited. I was going to be working on these weta, these animals that could survive freezing. I was going to be working on these cockroaches, and it was in the mountains, and it was just super great. It was super exciting. So I borrowed the department vehicle, the department four-wheel drive, to go off and go and do this do this field work and catch my animals so I could do physiology on them. And that's how the day ended, which is this is the department's brand new four-wheel drive, stuck up to both axles at 1,300 meters in the mountains, with a technician bringing his own personal Land Rover to come and tow me out of the thing. So I assure you, everything got better after that. And the thing is that, over the years in this department, they do a lot of field work. There's been a lot of people, write these vehicles off, have all sorts of bad accidents and things like that. But this is still the picture that they use when they advertise the health and safety training course in the department. So although you may be forgiven for your mistakes, sometimes they're so big that they're not going to be forgotten. So it did go a little bit better, and we ended up being able to go out in the winter and catch cockroaches and measure things and freeze stuff. And here's another piece of luck, was that just by chance, I ended up surrounded by a bunch of really, really ambitious and motivated fellow undergrads. So here we all are at in someone's apartment, kind of talking, and these are the honors thesis students from my year. And the thing that's crazy is we all kind of ended up in science one way or another. So Lisa did her honors project on viruses in lakes and is now an ecological consultant and a teacher working mostly on viruses in lakes. Darren here he never actually did a PhD, but he's ended up working. He's doing his project on systematics of these little weta grasshopper things, and he's now an entomologist doing systematics for the New Zealand government. Margaret, who's married to Darren, by the way, she did a PhD in bird behavior, but she's now a professor at Auckland University studying biosecurity and border controls of invasive species. Simon did a master's on blood parasites and ended up working in corporate finance. We don't talk about Simon. And Ross was working on stream food webs. He did a PhD on stream food webs, and he's now actually the director of the Institute for Applied Ecology at the University of Canberra. So this was kind of pure luck that I ended up surrounded by these people that were super ambitious. And this wild thing happened, which was that Darren was doing this systematics. He was identifying these species. And it turns out that the group that he was working on was taxonomically a colossal mess. And so after he'd been, faffing around for about 3 months, he realized that he now had a better key for all of the species in this genus than anybody else. So he submitted it for publication and it got accepted. And we were like, I want some of that. So it led to this kind of kind of motivating force. We were all really interested in publishing. We were all really interested in framing our research as research that was actually publishable research. And we all got publications out of our honors thing. I was lucky that there was this ecosystem of people. So with Bill Block and Roger Warland, we wrote a bunch of papers, and I was able to get a couple of papers out of my undergraduate thesis as well. So the point here is that undergraduate research is real research, right? Just because you're learning on the job doesn't mean that the research you're doing can't actually be publishable, can't contribute to society and to science. Now, don't get me wrong. I am not Don Griffin or Marla Sokolowski. My undergraduate work has not had the kind of impact that theirs has. But, and I see my master's student Enrique down the back there, one of the papers that I'm really proud of is actually one that was about whether or not ice forms inside the cells of these freeze-tolerant weta. And I still think it's actually a really good paper. It's one of the few that I go back and like actually kind of wish people read and cited more often. So in the process of doing this, I learned how to design experiments and ask questions. I did a lot of writing, and you have to realize that in science, we are basically professional writers that do a bit of science on the side, because if you're not communicating your research, then it basically doesn't exist. And I learned a lot about how to publish and publishing norms and expectations, which was really useful at the time and completely useless now, because at the time to publish a paper, I would print it out and pentuplicate and stick it in an envelope and from New Zealand mail it to the other side of the world, where it would be put into some kind of, cement mixer and sent to reviewers and come back, sometime in the distant future. It's much faster, or usually much faster now. And then this early success then leads to scholarships and grants and so forth and such like. So success begets success. And that goes for any undergraduate research, that if you can do research that involves you in publishing a paper or being involved in a conference presentation and things, that is then seen as sign of your potential and as an is used as evidence that you are worth investing in the future. And these things kind of snowball a little bit. And I think the most important thing is learning by doing, right? There's no situation, I think, in anything where you do all of your training and all of your training and all of your training until you end up perfect at it, and then you get to do the real thing. You always learn by doing, and by doing undergraduate research, ideally with involved people and with graduate students in the lab and things, you get the opportunity to actually make some mistakes, learn from those mistakes, and actually learn by doing real research. This also led to a bunch of lifelong interests. I didn't grow up in an environment where I ever went outside or went, or did anything, really interesting or traveled. And it really opened up like a whole bunch of opportunities. I met different people. I got involved in different things, got involved in all manner of, sort of dangerous outdoors activities and things like that, which I just wouldn't have done just growing up in the suburbs of Auckland. So if I can summarize my path here, we have undergrad classes, a field course, we have some volunteer experience, ended up with some paid experience and an honors thesis. A bunch of serendipity, so the topic in class, right place, right time, happened to be with a bunch of other people that were motivated as well, and we fed off each other. I developed a genuine out-of-class interest in the topic, and that was important because it meant that I developed a good understanding. And I think that anyone that is making use of their opportunities in research is really synthesizing their experiences from lots of things. So it wasn't just that I was paying attention to those 2 lectures in that class. I was using the other things I'd learned in that class and evolutionary biology classes and ecology classes and things to actually put together a story. And I had camaraderie, we had a little bit of competition, and we had this drive to publish, which was super useful. I think, though, that the most important ingredient is curiosity. You've got to have to want to know stuff. Right. And this is just a quote from Knut Schmidt-Nielsen, which I quite like, which is that the primary function of our schools is to impart sufficient facts to make the children stop asking questions. Okay. Those with whom the schools did not succeed become scientists. Right. So if you ever explain anything to a scientist, one of the things that you notice is that they always have a whole bunch of other questions about how it works and why it works, and and why you think that, and whether the evidence is really any good. So along the way, I'm not going to talk about the rest of my research career because that's like not undergrad, that's not relevant. But I did have the opportunity to influence a bunch of undergrads along my journey training before I landed here. And I'll talk about the undergrad research that we've had here. And just to shout out to 3 of them, because I'm particularly proud of these individuals. So one is Caroline Williams, who was an undergrad in a lab that I TA'd in 2001. And she ended up actually doing a master's with me as a kind of a co-supervisor at Otago, came here to do a PhD, and she's now a professor at Berkeley and is producing like grand students. Like she's graduating PhD students who are therefore my kind of my academic grandchildren. Natasha Motapo was an undergrad who was helping me out in the lab, and I was able, and she was really interested in science. She was interested in insect physiology, and she ended up doing a master's and a PhD on ants. And she's now the director of one of the postdoc support office at the University of Stellenbosch. And a third person is Terry Nielsen at the time. So she was an honors student in Las Vegas at UNLV while I was there, and she did an honors thesis and did actually a really nice study on how carbon dioxide affected cold tolerance in Drosophila, and then went off to medical school, as I'm sure many of you are planning to do as well. A year or two ago, she messaged me and just sort of said this, and I thought I'd share it with you, which is that people, and by that she means patients, she says, still ask me about our work together since it pops up when they Google me. So she's working as a nephrologist, and so people are like, oh, who is this person that I'm going to see? And they Google, and they're like, the various hospitals she's involved with. And like this one entry in the scientific literature of like Drosophila and carbon dioxide and cold. I really learned a lot. I'm a nephrologist now, and physiology is the main reason I picked it. So even though she's not really working on insect cold tolerance anymore, understanding and thinking about physiology has turned out to be really important for her work. So thanks for inspiring me those years ago. So the things that we learn by doing research are things that you're learning as undergrads in lots of different places. Right, so you've got your course skills. You do your stats course, which I know you've all forgotten by now, but it really was important. There's a reason we make you do it. You've got experiences, so field courses, volunteering, work study, USRAs, internships, whatever. Synthesizing your knowledge, realizing that like your different classes don't exist independently, but the information in them and the content and the concepts cut across the things. Curiosity is super important. Reading more broadly and deeply than you are required to. And then a couple of other things. So building your network, adopting mentors, and seeking advice. And that's something that I can't emphasize how important that is and how easy it is to do. Because if you look around, you've got sort of 50 or 60 people here who are the beginning of your network in science and outside of science. And then, of course, communication, writing, numerical communication, and verbal communication. So that was quite a long introduction to me and my path. And so now I'm going to do a bait and switch, because I realized that I'm not really going to talk about insect cold tolerance as much as I'm going to talk about insect physiology, because I chose a few topics and they're not all about insects and cold. And the way I'm going to do this is I'm going to talk about four different papers that I've published in my time at Western that were led by undergraduate researchers, mostly honors thesis students. So the first one is by this person. I seem to have terrible photos of all these people. I don't know what it is about me that I managed to either not take photos or lose them of people. So these are little blurry things from the photos on my website of the group at various stages. So Amy Bazinet did this work on a thing called rapid desiccation hardening. Right. And so essentially what this means is that you have, this was in Drosophila, but many insects will do it, which is that if there's a sudden change in the kind of desiccation stress that they're exposed to, they need to be able to modify their susceptibility to that desiccation stress really quickly. And we know very well that this happens with cold and with heat, but we don't know much about what happens with changes in water balance. And so there was an old paper at the time, about 25 years old, that suggested that Drosophila might be able to do this rapid change. So we kind of threw the idea to Amy and provided some suggestions. And so the first thing she did was she showed that rapid desiccation hardening happens, right? And she showed it in males and females. So here we have, so here we have males and females. Oh, this is actually, I'll talk about this in a bit. Basically what happens is if you expose flies to a short period of dehydrating conditions, give them a bit of time to recover, then afterwards they will have a very big shift in the amount of water loss. So our first hypothesis was just that they changed the fuel that they use. So one way that insects can store water is by using glycogen as a carbohydrate store, but using it to act as a big sponge, right? It has all these hydroxyl groups. They make friends with all the they make friends with all the with water molecules, lots of hydrogen bonding, and so that water is not osmotically active, which means you can store water without diluting your blood so much that you die. So then if you want that water back, all you do is you start using glycogen as your fuel source. You break those molecules up, the water molecules fall off, and you can increase the amount of water that's available. And that's not what flies do. Sorry, that was a long way to say we tested this hypothesis and it wasn't true. What it turns out, so she then did a bunch of really complicated experiments measuring the amount of water loss in real time in these flies. And what she was able to show is that the hardened flies, and the open boxes here, both males and females, what they do is they actually reduce the amount of water that is lost through the cuticle. So they've actually changed the waterproofing of their cuticle in the space of about an hour, from being not terribly waterproof to being a little bit less not terribly waterproof. I mean, they're still not like completely bomb-proof animals. Another couple of undergrads a bit later on, Joe Stenziano and Richard Sauvé actually then showed that the way that this changes is they actually change the layer of hydrocarbons on the surface of the cuticle. So basically they secrete new waxes that make the cuticle more waterproof. Okay, there are no good transitions between these papers, by the way, because I just chose them as being nice things. So, yep. And now we're going to talk about Sarah Lake. So Sarah was an honors thesis student and just over a decade ago. And she was a very different kind of experiment, because what we knew is we were very interested in the thermal sensitivity of metabolism. So ectotherms usually have a relationship like this between temperature and metabolic rate. It's curvilinear. You've seen this in 2601. You will see it again in 3601. You'll see it in 3602. You'll see it in 4602, I promise. And so the thing is that it's useful to be able to measure that and estimate it, because we're really interested in how it changes in different kinds of environments, and as a way to reduce the amount of energy that animals are using in the winter. And the thing was that there's different ways to measure it, and they are differently hard and differently time consuming. And so what Sarah did was she took all of these different methods, did all of them on lots and lots and lots of crickets, to show that it didn't matter which one you did, you could use them all, right? So in some ways, sometimes when people present their research, especially students, when you're analyzing your research and you're doing an analysis and you're like, oh, and it's not significant because you did your stats course, and you get like a p-value and it's like, it's not significant, and you wanted to do something significant, and it's very sad. However, in this case, we were ecstatic that we were not able to find differences between them, because it meant that you could use whichever method you wanted, and some of them are much easier and cheaper than others. And so this becomes a really important paper for my lab and for others, because it means that we know that we have quite a lot of flexibility in the way that we design our experiments. This is a double duty honors thesis paper. And so Claire Newman was mostly actually in Tony Percival Smith's lab, but also kind of hung out in my lab. And a bunch of my students and postdocs and some collaborators from other places, and Julia Ahn, who was a 4970 student. And this was all about this gene called FROST. Now, as an insect cold tolerance person, how could I not love a Drosophila gene called FROST? Obviously, this is the gene that I should be studying. And the reason that it's obviously the gene I should be studying is that when when you expose a Drosophila to cold and give it a little bit of time, FROST is like the one gene that is massively upregulated. So it looks a little bit like heat shock proteins at high temperatures. So this is pretty amazing. Cool. It's all about FROST. And so the trouble is that we couldn't really figure out what it did. And we did various experiments that suggested that maybe it didn't do very much, which is a bit awkward. And it ended up in this situation where Hiroko, who was a postdoc, had done some work using RNAi to knock down FROST expression, and we found that it had no impact on the cold tolerance of the flies. But then a couple of my friends in Australia used RNAi to knock down FROST, did exactly the same experiment, published in a much better journal, and said, look at this, it does affect cold tolerance. So we're like, well, that's no good, because obviously we're right. But so how do we prove that we're right? Well, the way that we prove that we're right is by actually making the first use of CRISPR in comparative physiology, which was to delete frost entirely from the genome. And what we could basically show was, so here are our various, so what we were able to show, oh, I've unplugged my side, that's okay. There's plenty of electrons in that computer now. So what we were able to show was that it made no difference to the cold tolerance of the flies, but it made a big difference to the reproductive output of these flies afterwards. So these colored things are the various CRISPR deletions and insertions and things that mean that frost doesn't work. And the black bars are our kind of wild type. And what you can see is that every time you stop CRISPR from working, if you expose the flies to cold, then they don't produce as many offspring. So unhappy flies only if they're exposed to cold. Associated with this, we also started to wonder, well, what is it doing? And so we did a bunch of work to try to figure out where frost is being expressed. And we were able to show that it was in the abdomen. And then Julia and Hiroko dissected like most of the world's supply of Drosophila melanogaster to show that it's actually specifically expressed by the Malpighian tubules. And what I've been asking Tony for Christmas for 1,000,000 years now is a version of frost that's labeled with GFP so I can see where that protein is going, but we don't know that yet. So, last paper that I'm going to talk about, I think, is this guy, Adam Smith, and also Julian Moulton, who was an undergraduate student from Colorado College who came to visit for a summer. And that's something to bear in mind, is that you don't necessarily need to get all of your research experiences right here. There are opportunities for exchanges, there are opportunities to travel, there are opportunities to volunteer in places that are not here if you're interested in getting involved in research. And anyway, and so this is the stuff that we work on now, which is a cricket called Gryllus velitus. Am I going to find them if I look inside this box? I am. Gryllus velitus. Behold, they're magnificent, I promise. You can see them later. And the thing about Gryllus velitus is that it's a cricket, it's the Springfield cricket. These are the guys that you hear chirping away in kind of June-ish, right? So they overwinter as a medium-sized baby cricket, the 5th instar nymph. And in that state, if we treat them right, we can make them survive freezing. We can make them freeze tolerant. Now, something we've known for a while is that if you combine two of my favorite things, you freeze stuff and you do it inside a respirometer where we're measuring carbon dioxide production to estimate metabolic rate. When the animal freezes, you see this big burst of carbon dioxide. Where does it come from? And it could be a couple of things, right? It could be that it's from it could be that it's because there's a metabolic response to freezing, or it could just be that there's some other source. And so we can cool our crickets down to -8. Some of them don't freeze, some of them do freeze. So we have this nice comparison. And what you can see is this is temperature here. And the colored things are the CO2 production. And what I want you to see is that there's this little burst here, which is the CO2 that's produced, and a much bigger burst there. And so what this told us was that freezing was producing CO2. And then through a bunch of clever experiments, knocking out different parts of the metabolism of crickets using targeted insecticides and cyanide and all sorts of other fun and lovely and evil things, we were able to show that actually this CO2 is not metabolic at all. It's just the stuff that's hanging around in the insect's blood anyway. When you freeze that, there's no space to keep the CO2 anymore. It comes out of solution and gets released from the cricket. More interestingly, we get this huge burst of CO2 when they thaw out again, which suggests that it's really expensive to thaw out. And what we realized is that this doesn't include all the space in the hemolymph now for additional CO2. So there's like a whole bunch more metabolism we're not getting. And then the other cool thing is that this is what a normal cricket looks like. This is its spiracles opening and closing, so they breathe through little holes on their sides. And what you can see is that it takes a good few hours before our frozen cricket actually recovers nervous activity enough to be able to open and close those spiracles again. Okay, so each of these projects was a lot of work. Each of them was more than the minimum bar for an honors thesis project, right? So In each of these cases, the student did that work because they were motivated by the question in the project and because they were excited about learning something new. And I don't just mean something new to them. They were learning stuff that was new to science. They were seeing things that no one had ever seen before. This meant that they were happy to do things like repeat experiments, add controls when we realized we hadn't entirely accounted for something that we thought might be important. And they went well above and beyond the work that they wrote about in their proposal. Along the way, they learned a whole bunch of things: lab skills, communication, persistence, precision, and project management, right? And if you were out there in the real world hiring someone, which of those skills are you like, I have no interest in those skills at all? These are all like real world valuable skills. And in fact, of the people that I've told you about, only Julian is actually still is actually in academic science. He's about to start a PhD at Riverside. But everyone else is a lab tech. Sarah works as an executive in the pharmaceutical industry. Julia went to dental school. Claire went to genetic counseling, and Adam is like now working at Bayer as a science rep. So the point is that they got skills from their honors thesis, from working hard, from doing real research that has served them well in jobs that are not necessarily in academia. So just because you don't think you want to be a professor doesn't mean that getting involved in understanding research isn't valuable. Now, I think it's important though, because I've been telling you these stories about, people that have these like, road to Damascus moments in third year biology and go on to like devote their lives to a single gene that they found in like one maggot crawling around in a petri dish. And it's important to realize that this stuff doesn't have to be a passion project, right? You don't have to be like, I am waiting for the one thing to whack me between the eyes that I am super passionate about. And if you are passionate about like a subject, I'm passionate about genetics, you probably need to think about what that means, because it's different from just doing well in the class. It's actually being interested in the topic. However, I don't think you need to like wait for the passionate thing to hit you between the eyes. So the quote that I use here is from my wife. Here she is when she was a PhD student in South Africa. And she did a really cool PhD on these birds. This is one that's upside down being weighed in this high-tech plastic bag. See, it's got a little hole at the bottom so it can breathe. And the interesting thing about them is they have these ridiculously long tails. So she was doing this experiment, manipulating tail length and looking at what that did for the number and quality of offspring that they fathered. And the thing was that Vari is interested in lots of things. And to be honest, tail length in sugar birds was like fairly low on the list, but it was a great opportunity. Right? It was an opportunity for her to go and do a project where she could like learn new things and do cool stuff. And as she said, she's like, if you're good, you become passionate about the problems and opportunities that come your way. She did a very good PhD. And it's not because she, when she was a five-year-old girl, she was like, I want to cut the tails off birds. It was because she realized like that she could learn cool things and she could do useful things and contribute to science and society along the way. So it's important to understand that your undergrad research doesn't have to define you. So I've made a list of various people here, and these people are not normal, right? And I want to illustrate that. Actually, this is just a photo. This was quite a good one because we had quite a lot of people associated with the lab over Zoom during the pandemic. Whole bunch of people there, all like graduate students or whatever, a few undergrads as well. And the key thing here is that not many of them, the honors students are not now working on insect cold tolerance, and the people that did do honors that did come to do grad work on insect cold tolerance, most of them had done honors projects on all sorts of other weird and wonderful things. So your honors project doesn't define you, and you have lots of opportunities to change directions. The exception there actually, so there's me and Jackie Levinson, who was a volunteer in my lab about a decade ago, and she graduated with a PhD from my lab just earlier this year. So I went through these skills, and the key thing is that these are not just skills for research, they're not just skills for doing real research, they're actually skills for jobs as well. And there are ideal paths to success in science, right? So, so here's Heath McMillan. He was in your position. He was an undergrad at Western. He volunteered in my lab, did a field course, did a USRA, did an honors thesis, did a master's and turned that into a PhD, co-supervised by Jim Staples. did like a postdoc in Denmark at one of the best comparative physiology departments in the world, got a Banting postdoc, which is like the absolute premium postdoc at York, and is now tenured at Carleton. You can actually see him give a talk later this week. Quick plug for Heath, BGS 0153, he'll talk about microplastics. And Jackie, I just talked about, same deal. She like kind of came through the lab and is still doing what she was doing. Now, that is still not normal, right? And there was also a pandemic, which does kind of put a little bit of a hiccup in even suggesting that that's the path that you should take. So I'm just going to kind of make a quick list of how I think you should approach this. If you are an undergrad and you're interested in research and you want to go where research might take you, some ways that you can approach this. So if you're in first year, talk to your TAs, see if you can get into a lab as a work study student, as a volunteer or something like that, right? Just to like roll your sleeves up and get some cricket goo or bird food or whatever under your fingernails and start to learn a little bit about what happens in a lab. Because when you're in a lab, sure, you get to like wash lots of dishes and things, but you also get the opportunity to hang out with the grad students, to talk to them about what went wrong in their lives that they're at grad school, and how to go about getting more involved in science. If you're in second year, all those things again, but you can also apply for a USRA. You can do a field course, right? And I'll talk about the field courses in a moment. And then once you're in third year, you need to start thinking about if you're going to do a thesis project, right? But you can also consider the science internship program. And you'll see advertisements for that, I think, at the moment, right? And you sort of need to get into it around now to be able to do it between third and fourth year. And if you're in fourth year and you're not in 4999, then you still are probably going to need to get some lab experience, and you're going to need to develop your thinking skills and reading breadth. And there's a few ways that you can get some lab experience. Quick plug for field courses. So this biology is part of the Ontario University's field biology program. This is an amazing program. There are a whole bunch of universities that run field courses in Ontario. And essentially we get the places in that program that we provide. So we have a couple of field courses that run here. Are you going to Belize, Graham? Yeah. Graham is, yep. Graham Taylor here is leading a trip to Belize. There's and an Algonquin Park one. There's Paul Mensink runs a course out at Huntsman Marine Lab. And anyway, but you don't have to. You don't have to hang out with Graham in Belize. Some people don't want to like hang out around the coral and the boring fish and all those things. But you can enroll in any of those programs, right? There is some money. The application deadline has passed, but you can still apply. It just means that you won't necessarily know that you've got the support by the registration deadline. In theory, that QR code will take you there, but if you just Google Ontario Field Courses UWO, you'll get to approximately the right place to find out about it. Quick plug for the science internship program. So these are paid positions. They're basically the same as like a co-op at Waterloo or something like that. There's up to 16 months of paid employment, often in science. We've had a bunch of people who have done this that have worked in my lab at various times. So Adam, I talked about his research. Kevin is now a data analyst. Lamise, give us a wave. Where are you? I saw you there. There's Lamise at the back. So she did an internship in my lab, and she sort of took a little bit of time away, but basically we won't let her leave really. And another quick plug is that I have an OWL site and some meeting slots and things to talk about what happens after undergrad in terms of how you apply for master's degrees, how that works. I'm making little videos so you don't have to, or you can listen to me at your leisure or on mute. And a bunch of other resources about how to go about getting to how about getting to grad school. I think so if you hunt around and get to a bit where there's joinable sites and you search for biology graduate school prep, that sort of should get you there. Otherwise, send me an email and I can add you manually. So moral of the story, undergraduate research is real research and you can do it. You have to be curious and you have to work hard, but you don't have to be super smart. You don't have to have amazing grades. And if you get involved in it early, the more you learn, the better you'll get at it. So I made a list of all the undergrads that have that I've published with over my career since I've been a professor, and I'm very proud of all of them. I believe that I'm very excited about the QR code thing. You know, it's like it's nearly as exciting as like a couple of years ago when our lab discovered this amazing new tool called the microscope. So that should take you to our lab. Various institutions I've been involved with, and thank you very much, Bugs, for having me. And I hope that this is useful. I can take questions, or you can drop me an email, and we can continue the conversation. Thank you. So I guess one thing that, you know, it's sort of going forward with science, always thinking ahead to the next thing. And so you mentioned sort of supporting people thinking about grad school. When should undergrads ideally be contacting people or acting in some way on grad school? So this is so the question for those of you online is from Hugh Henry, who is exposing himself as a former grad chair in the department, because his question was, when should someone that wants to get into science be thinking about going to grad school and contacting people? And the answer is right now. And it almost doesn't matter if you're in first year or in fourth year, but now is a really good time. If you're in fourth year and you're thinking about doing a master's next year, you should probably really be on the way to thinking about it. But even if you're in first or second or third year and you think that this is a direction that you want to go, start asking your profs about it. Go to office hours. Office hours need not be a period of quiet, solitary reflection. You are allowed to come and talk to us. And you can actually start to get ideas. And just because you talk to me about going to grad school doesn't mean that you've suddenly signed up to do a master's in my lab. What it means is that you started to open that network, which means that when later on you have that really interesting lecture about naked mole rats, and you can be sort of like, hey, Dr. Sinclair, like, I think I'm really interested in naked mole rats. How, what? And I could be like, oh yeah, like I know a couple of people that work on those. You can contact this person and that person or whatever, right? So this is how your network works. If you're in fourth year, you do need to start thinking about it pretty soon, but there's lots of flexibility. There's lots of little outs and things. And some of the videos that I'm making and posting on the site sort of talk about some of the ways that you can manage that. Yeah. Is that reasonable? We've got the previous grad chair and the current grad chair. Did I say anything that you'll regret? No. Okay, good.
You don't have to start right after fourth year. Take a year off?
Absolutely. Take a year off. That's what my wife did, actually. She took a year off and traveled the world and did cool things. Yeah. Yeah. You with the magnificent hair.
So you talked about building a network, and I just was wondering about during the pandemic, because there's so many things that are online, it becomes really difficult to build that network and have normal interpersonal connections, because sitting in a room and having a lecture like this is very different than sitting on Zoom. Because here I can decide to be like, hey, what does Belize look like? Or whatever. But yeah, it looks great. But on Zoom, it's very, very difficult to just kind of stumble across somebody. So what would you recommend for students that have really know nothing except for that online platform, and how to make connections when you are really just looking at the screen?
Yeah. Okay. So, sorry, Sam, do you? Yeah. Yeah. Yep. Totally. Yeah. So for those of you online, the question is, if we've just come out of a pandemic where all of the human interactions have been electronic and on Zoom, how do you start building your network now, right? And so, well, all of you here in person are doing it right, and people on Zoom, you need to think about coming to in-person things when you get an opportunity, right? Because it is much easier to build those relationships in person. So places networks, research networks come from for you as an undergrad. Like I say, coming to office hours, talking to profs, getting to know them, really useful. Talking to your TAs, like your TAs are actually amazingly, they're people, or most of them, and they're actually quite excited to be there teaching you. And they are graduate students and they've seen a bunch of stuff. They might have come from different institutions and things like that. So get to know your TAs and ask them questions about their careers and things like that. So that's like some initial ways to start building your network. Doing bug stuff is a good way, because some of you are going to end up discovering that later on, these other people are also doing research. And then if you can get yourself involved in a lab one way or another, then that's another good way to start building relationships with some of the grad students and postdocs and various other people. So, but I think you're right that like personal interactions are important. Field courses are great for getting to know profs, because one of the things that you'll need when you're applying for things like grad school is you need profs to actually write things about you that don't just say, Brent was a student in my large second year class and I couldn't find any complaints in my email, so he must have been okay, right? So you want like something more than that, and the way to do that is actually to be involved and to do things and give profs an opportunity to see you perform as an intellectual, three-dimensional human being. Yeah, does that help? Okay. Yes.
So kind of adding on to what she said, and like more specifically with like the office hour aspect, like do you have any tips like specifically towards that like first approach and like how exactly to like talk to a prof or a TA about like research or grad school, like what specifically, like how to approach that?
Okay, so the question is how to approach it. Okay, so everyone stand up. We may as well make this interactive. Everyone stand up. Okay, now repeat after me. Hi. Tell me about your research. I'm really interested. Okay, you're all perfect at it. So seriously, like especially grad students, you can sit down again. Sorry. So seriously, we like talking about research. We're much more interested in talking about our research than we are about what was on like point 3 of slide 6 of lecture 9. And and you can ask questions. You can ask like, how did you get into research? Why are you doing what you're doing? What are the good things and the bad things? What should I do if I'm interested in it? Or I'm not interested in what you do, but what should I do because I'm really interested in seals or banana slugs? So there's lots of so you can talk about that. But there's a few people that TA there, like Zach and some of the people at the back. If someone asks you about your research, you're kind of excited by it, right? Yeah, they're like, oh my God, here's an undergrad that thinks I'm an actual human. That I might have a life, that I don't just sleep in a coffin in between like lab sections. So absolutely, it's not that hard to do. So yeah, on the OWL site, and I haven't quite made it yet, there will be a video about how to initiate a conversation with some prof that you found on the internet or through the web of science or something that you think you might want to do a master's with. And I've got a bit of information about how to initiate that interaction as well. But that's a little bit more formal. If you can rock up to office hours or talk to a TA during the lab, then that's by far easier, and you can do it fairly informally. And honestly, like I sit in office hours all the time. It's period-required solitary reflection, and on occasions when people come, you don't have to ask just about the course. I'm happy just to, the time is yours as part of the time that is allocated to the course. So make use of it. Yeah. Okay. Oh, yes.
Sorry, I do have another question.
That's okay.
It's a little bit unrelated to research in general, but I found it very interesting that you mentioned that originally you wanted to become an opera singer. Personally, I'm taking a double major in classical music as well, and I was wondering how you navigated balancing science with having another passion, if you did choose to continue with that.
Okay. Right. So I've been called on my claim that when I was 16, I wanted to be an opera singer, which is true, right? However, although I had the muscle for it, I did not have the technical background or the application that I was going to take it as far as university. However, I did start my undergraduate degree doing a double degree, not a double major. So it was going to take like 5 years and be horrendous in English literature and philosophy and biology. And so, and this is not going to be a good answer for you because it's very hard to navigate that. And part of the reason is that all of my biology classes had lectures in the morning and like 3-hour labs in the afternoon that filled up the entire afternoon. And all of my English literature and philosophy classes were English literature and philosophy. So of course they were in the afternoon because, like, you know, humanities people are like sleep in coffins during the morning. So essentially I ended up with these like horrific timetable clashes, which is why it was going to take 5 years to get these 2 undergraduate degrees. And so one of my summer jobs was, quick aside, anyone that's had a pool, you know, you buy pool chlorine and it's in these like plastic tubs and it's got a lid on it. So my job, I was like the guy that would weigh the weigh the chlorine into the pot and then bang the lid on with a rubber mallet. So so this is not my first like long period of time wearing a mask. And so there was me and this other guy, and we would sit there in this like super hot shed because it had to be away from everything else that might explode, and pour these things on and bang the lids on the pots and talk through these like really like dense respirators. So hey, the conversations like this all the time. Anyway, and I was trying to like figure out my timetable, and he was like maybe a couple of years ahead of me in undergrad. And I was like, so I got this real problem that it's really hard to fit my biology courses and my English courses in together, and I'm not quite sure how to do it. And he was like, oh, well, like, what are your marks like? And I was like, well, you know, my marks in biology are really good, but that's because biology is easy. And my marks in English are okay, but that's just because it's like challenging and interesting. Bang, 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 poor, bang, 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 poor. You know, and he's like, well, has it occurred to you that maybe you're good at biology and not very good at English? and this had not occurred to me and was an absolute revelation. And so I actually ditched the English degree. And the thing is that I don't think I needed to spend 5 years as an undergrad getting both those degrees. And I don't think that not having the English degree has in any way diminished my interest in, or the philosophy degree has diminished my interest in critical thinking, in literature, in you know, reading and writing. So you don't have to get an undergraduate degree in something to enjoy it and to do it well. Having said that, and I'm the wrong person to do this because like I'm not technically competent at music, so I can't like play an instrument or anything like that. There are a lot of scientists that are also dedicated and sometimes very good musicians. So that's not it's not an unusual path. Yeah. So yeah. So and have other interests anyway. It's nice to have a life. Yeah. Okay. So I've been talking for a long time here. It's been over an hour. Are there any questions online that you want to relay or anything, Sam? No, all the people online are asleep. So I'm going to call it now if people want to talk to me more. And we have some insects here, which my minions will help you to handle if you want to meet some of the insects. I didn't talk about insects probably as much as I should have done. And if you want to talk to me or you want to sign up for that course and the QR code didn't work, which it probably didn't, then drop me an email and I'll sign you up for it. Okay? Thank you very much.
Thank you so much for agreeing to.
Thank you so much for inviting me.
It's really nice to see that there's so many people who are also really excited to undergraduate research.
Yeah, it's really important. Like it's like, and I think you can learn a lot from it. Yeah.
Well, so like, should I ask if they want to sit?
Yeah, absolutely. If anyone wants to play with the bugs, they're here.
They're African.
Don't you see them like normally they're.
For the
No. Okay, oh, gee.